This is a production of Cornell University.
So, as Chris said, I am a former graduate student from Cornell, and looking back on my career, those were some of my favorite times. The times when I lived in Ithaca and then up in Geneva with Harvey Hoek as well. And I got married in Geneva. My wife and I got married on the lakefront up in Geneva, and we look back on those times as being some of our best friends and some of the best times of my life. So I just want to say, especially to the students of you who are there, you are so fortunate to be attending school at Cornell. It's really a fantastic place. Okay, so if you know my career at all, you probably think of me as an Aspergillus nigilans geneticist. And much of my career, I have focused on that area. I'm looking at hyphal growth, but I'm actually going to tell a different story today because I've recently become interested in particularly Colletotrichum graminicola. So I am a microscopist and a cell biologist, and Colletotrichum really has amazing cell biology. It's probably even better than Neurospora. If you're in the field, I think Colletotrichum, there's a lot that can be done with Colletotrichum. But anyway, I'm going to tell you about a story with Conidia, the Conidial Cointos. And we can see in this image two different Conidia depicted. And I'm trusting that my mouse that I'm moving around here is visible to you all. If it's not, then please somebody yell at me so I can figure out how to fix it.
Looks good.
Okay, thank you. And there are two Conidia here. One of them is stationary, and therefore I would call it attached. The other one is not stationary, and you can see that because subsequent pictures that are depicted in this are showing a blurring, particularly at the tips of the spore. And so this is where this concept of the coin toss comes from, is that there is roughly a 50-50 selection mechanism for the attachment of these spores to their substrate. And so I'm going to tell you how we got to that conclusion in today's talk. Okay, so as most of you are probably aware, Colletotrichum graminicola is an ascomycete that causes various diseases of corn. It's one of the top 10 most economically relevant fungal pathogens. It causes foliar lesions on corn plants, but also is associated with stalk rot, which causes the lodging of the stalks and makes it impossible to harvest the corn, so it's very economically damaging. It's one of the greatest sources of yield loss among maize pathogens, resulting in tens of millions of bushels to be lost each year. And I pulled this image out of a review article by our very own Dr. Bergstrom. Okay, so my interest is in the early events of the production of spores and in spore germination. And so let's take a brief overview of what happens here. So Colletotrichum produces two types of Conidia. There is a micro-Conidia that are often called oval Conidia in the field that are not going to be part of this talk because they're not part of this mechanism that we talk about. Oval Conidia are thought to be involved in the dispersal of the organism within the plant. But the dispersal outside of the plant and to the plant is mediated by the macroconidia, which are also called falcate conidia due to their shape here. And those are the spores that we see in this image. They're these crescent-shaped spores. When the spores germinate, typically you'll see one germ tube emerge that very quickly, under appropriate conditions, very quickly that germ tube will stop polarized growth and begin to differentiate the infection structure, the Appressorium, as we see in this image. And then it will penetrate its host using a melanized Appressoria, using a mechanism that is very similar to the better characterized system of Magna Portho or Pericularia oryzae. So it uses melanin-derived turgor pressure in order to penetrate its host. And sometimes you can get septation in the spore in order to get multiple Appressoria forming from the spore. And then once it penetrates the host, you'll get ramification of hyphae throughout the maize tissue, as we see in this image, where the maize tissue is stained red and a GFP expressing strain of Colletotrichum shows us where the hyphae are growing through the infected host. Okay, several years back, I was, along with a grad student, a former grad student of mine, Chi-Li Wang, we created a Life Act strain of Colletotrichum. So Life Act is a reporter that shows us in live cells the actin cytoskeleton. So these next few images we're going to see are showing us what the behavior is of the actin cytoskeleton during these early events of germination. So we see up here a dormant spore, pre-germination spore, that already has this well-defined actin cytoskeletal network running from tip to tip. As the spore germinates, and here I'll be showing a time lapse, and this is sort of setting the stage for the types of data that I'll be showing you throughout the talk. This is a time-lapse video where a picture is taken approximately once a minute and then is being played back at a much faster rate. So we're seeing hours and minutes represented in the video. But in this video, we see the germination of a falcate spore where the actin cytoskeleton is highly polarized. And I'll wait for it to loop back around again, and we can begin from the beginning of the sequence. We have a highly polarized actin cytoskeleton in the tip of the germ tube. That actin retracts to a septum as the septum is differentiated within the spore. And then once the septum is delineated, the actin then migrates back out to polarize in the growing germ tube. So that's germination. Here now we're going to look at Appressorium formation in a similar time lapse. These time lapses are approximately 3 hours long. So the septum has already formed in this time lapse, and we now see polarization of the actin cytoskeleton that then depolarizes as the germ tube begins to swell to form an Appressorium. And the actin all then relocalizes to the septation site as a septum delineates the Appressorium from the spore. And then the final time lapse in this sequence will be looking at an Appressorium from the point of septum formation to the point of penetration peg formation. So the actin was localized to the nascent septum, the septum forms, actin redistributes into the Appressorium, and now it begins to localize to the penetration pore as a penetration peg begins to form. So the point of all this is to show you that all of these developmental events require the actin cytoskeleton, and likely the trafficking of cellular components on the actin cytoskeleton to these sites of growth as the cell grows and differentiates. So my department just got a new building. About 2 years ago, we moved into a new building. And along with that new building, some of us were fortunate enough to get new equipment. And I'm no exception to that. I got a shiny new confocal microscope that has all of the bells and whistles. So if there is a piece of technology available on a confocal microscope, it's on this microscope. So this has really been a boon for my career since moving into this new building, because the types of questions that I can ask have greatly expanded. An example of that here is we see a growing hyphae of Colletotrichum graminicola, in which we're imaging sites of endocytosis. So each one of these foci are actually the sites of endocytosis. And unfortunately, I won't be talking about endocytosis in this talk, but that has been one of the focal points of my career for the last 10 years. But we have this nice new microscope that allows us to ask questions with even greater detail than we have done so before. So moments ago, when I was talking about these early events of germination, I showed you a dormant or pre-germination spore of Colletotrichum graminicola. This is the macroconidium or falcate conidium. And it has this pre-formed actin cytoskeleton that runs from tip to tip. Now, we noticed that when we did this study 5 or 6 years ago, but we were not able to go into any greater detail of what this preformed cytoskeleton might be doing. And the new microscope has allowed us to take another look at this. And so in this movie here that I'm going to show you, this is a Z-stack or a 3-dimensional projection of a spore, and we're rotating it so that you can see that the actin cytoskeleton is actually asymmetric within the spore. So it is on one face of the spore, not on the other face of the spore. And I have a visual aid for this. My visual aid is somewhat comical. It's this scrunchy banana, but it's essentially a falcate Conidium. And I trust that you all can see this. Somebody please shout at me if you can. But in this banana, which is representing the falcate Conidium, I've drawn with a Sharpie the actin cytoskeleton that's on one face. So in the image, we're seeing the spore rotating like this, and you're seeing the actin cytoskeleton is on just one face of the spore. So this was the first, when we imaged it this way, is when we realized that maybe there is something to this asymmetry. So we started to look at what could be the possible function or the mechanism for this asymmetry. Next, we decided to look at spores as they were formed on the Petri dish. So Colletotrichum, when grown on agar, will make these sort of erupent pustules out of their acervuli that has this gooey matrix in which the Conidia are secreted into from the acervulus. And so what we're looking at here are mature spores within that gooey matrix, just inverted onto the microscope so that we can see them. And I hope you will agree with me that the predominance of all of these spores have that actin cytoskeleton already made. There are a few instances in here, like maybe up here, where maybe you would say the cytoskeleton is not there, but I think that's probably a trick of the focal plane. So the actin cytoskeleton is pre-formed on pre-germination spore. Next, we looked at a young developing acervulus as it was producing the Conidia. And so in the center of this image, we have Conidia that are being produced and are still attached to the conidiogenous cells, giving rise to those spores. And they're revealed with a cell wall stain that shows a blue cell wall. And so all of these nascent Conidia that are still in the process of being formed do not have that cytoskeletal element. But all of the mature Conidia that have been released from these conidiogenous cells do have that actin cytoskeleton. So production of the actin cytoskeleton occurs early in the development of the spore, just after it is liberated from the conidiogenous cell. And to verify that claim that I've just made, in this sequence, we're going to look at a time lapse of a conidium as it is being produced. So in the image, we see the conidiogenous cell here and a young nascent conidium as it's being produced from that conidiogenous cell. And this image actually has 3 panes. We cannot see the GFP channel at this stage because the GFP is not being produced. So we have GFP, we have transmitted light, and then the overlay. And I'll play this image. So this is at 15-minute intervals for several hours. And you can see development of the conidium from that conidiogenous cell. As it reaches its terminal length, it begins to introduce that curvature characteristic of the falcate conidia, and we still do not see the actin cytoskeleton. But right about now, as the conidium is separated from the conidiogenous cell, you'll start to see that actin cytoskeleton form up. And you can then see the next conidium being produced from the conidiogenous cell concomitant with that. So I'll play that again. This is kind of an amazing feat of grad student ingenuity to get this image. This is the newest grad student in my lab, Ali Bedsole, who was able to get this sequence of a developing conidium of Colletotrichum graminicola. So still no actin cytoskeleton until the spore is released from its conidiogenous cell, and immediately deposition of that point-to-point actin array begins to occur. In this image, we are using an inverted microscope. So imagine that my finger is the lens and we're looking at spores settling down towards the lens. So we're watching spores settle. And as this loops through, you will see that some spores attach to the cover slip immediately. This one and this one both attach the instant they make contact with the substrate. But some spores remain unattached. You can see these continuously vibrating, moving around in a Brownian-like motion because they are unattached. And so this is roughly a half and half situation with spore attachment. And I hope you can also see that those spores that are attached have actin within the focal plane nearest to the substrate that we're imaging. And those that are not attached lack that actin cytoskeletal element in the focal plane that we're imaging. So when not agitated, we result in roughly half of the spores attaching to a substrate. And here that is depicted numerically in graph form. And this is kind of a complex graph. So I'll go through this. The gray line, the black bar graph is showing us the percent of spores that are attached over time. So 0 hours through 15 hours. And you can see that attachment hovers in the neighborhood of 40 to 50% attachment, no matter the time. And this is important that the spores remain undisturbed in order to see this 50% attachment rate. As soon as the spores are disturbed, the percent attached will change. On the line, we see the percent germinated. And so this is the next concept, that these spores must be attached in order to germinate. So if the spore is attached, then we achieve germination of the spore. So at 0 time, we've got 0 germination, but it's up in the 40 to 50 to 60% range over several hours after the spores become attached. But with the dotted line here, we see that germination does not occur at all on any spores that are unattached. And we've left this over the weekend, so 48 hours or longer on spores that are unattached remain unattached if they were undisturbed. They remain unattached and they remain ungerminated. Okay, so more pictorial evidence. Here we're looking at the same 2 spores in 2 different focal planes. So the first is the focal plane nearest the surface. And so you can see the actin cytoskeletal array running from point to point nearest the surface in the attached spore. And there's no observable actin cytoskeletal array in the unattached spore. If we go to the top focal plane of that spore, the same 2 spores, we have the actin cytoskeletal array still exists in the unattached spore, but it is on the wrong face of the spore. So again, I'll use my banana scrunchie as my pictorial demonstration just to make sure we're all on the same page. The Sharpie representing the actin cytoskeletal element. If the spore is on the surface on that face, it's attached, but if the cytoskeletal element is up, the spore remains unattached. Okay, and so here's some evidence for that in numeric form. If a spore is attached, then it likely has the actin cytoskeleton on the bottom nearest to the surface. If the spore is unattached, then that organized actin is at the top of the spore. And this is fully reproducible. And we do occasionally see attachment when the actin is at the top. And I'll explain that in a moment. There is a small amount of attachment using another mechanism that I'll explain when we get there. Okay, so this led us to make the proposal of the Conidial Cointos. And so here is a graphical representation of one of these spores with the actin cytoskeleton running tip to tip. And it wasn't until we thought of this in 3 dimensions because of our microscope that can allow us to achieve 3-dimensional imaging that we realized that that actin cytoskeleton is sided. So in this image, we're looking at the points of the spore facing towards us. So again, my banana, rather than looking at it like this, we're looking at it like this. And when this spore settles through water, these are splash dispersed spores, it's going to settle with the fat face of that spore down. And when it hits the surface, it rotates this way or rotates this way. And depending on the orientation of the actin cytoskeleton will determine if it attaches immediately to the surface or if it never attaches to the surface. Okay, so digging through the literature, we found a review paper by Dr. Bergstrom and Nicholson. And in that review paper, they addressed some older literature where the extracellular matrix of Colletotrichum was addressed. And so these spores they're liberated into a matrix on the from the Conidia cells. And so we're dealing with this matrix, which is this extracellular matrix, but also when the spores begin to germinate, they kind of secrete an adhesive matrix around the germline as well. And I think that that's what's being dealt with in this. But so it was previously characterized that the extracellular matrix is composed of glycoproteins, glucans, and amino acids. And so we scratched our heads a little bit about how we might be able to utilize this information on the microscope. And that led us to Concanavalin A, which is a lectin that binds to glucans. And so this is a protein that binds to glucans that can be conjugated to a fluorophore. So in this case, we used TRITC, Circie, which is a red fluorophore. And so ConA binds to glucans. And in this image, I'm using 3 colors. So first of all, I'm using a cell wall stain called solophenol that stains the cell wall blue so we can see the Conidia a little better. And this is actually a confocal-based fungal cell wall stain that Harvey Hope developed. So he was the originator of the use of this dye that's gaining widespread use in the fungal community. So here we have ConA, which is binding to glucans, and I hope you'll see that there is a strip of glucan on the Conidia. And when we then, with the other channel, image LifeAct, so the Actin GFP, you can see again our tip-to-tip actin cytoskeletal array within the Conidia. And then we do the overlay over in this image. And I hope you will agree with me that there is a one-to-one correlation between the presence of the actin cytoskeletal array and this glucan binding ConA reactive moiety, which we assume is an adhesive. At this stage, we assume is an adhesive. And this image, I think, is just absolutely dynamite because it shows spores in all 3 possible configurations with actin up, actin down, adhesive up, adhesive down, and then also the rare occasions when the geometry of the cell allows the spore to settle with the adhesive not in contact with the surface, but also not facing up. But again, the point of this is that we have a glucan moiety associated with the actin cytoskeleton. And here again is one of these 3-dimensional images through the spore, and then through the magic of computer assistance, we're rotating that 3-dimensional projection of the spore. And again, I hope you will see that secreted glucan, or the glucan moiety is directly correlated to the presence of that actin cytoskeleton. So our presumption would be that it's being secreted by components running on that actin cytoskeleton, but we have not actually proven that. And just to make sure, because sometimes seeing the video is not immediately obvious to everyone, I've got my banana, and what we're looking at is the spore doing this. And now the strip on the banana represents both the actin cytoskeleton and that glucan ConA reactive moiety. When I showed you the percentages in a graph earlier, I said that sometimes we hit numbers that aren't quite 50%. And I think the reasoning for that is actually revealed in this image, because the glucan moiety is also present at the tips of the spores. And sometimes spores aren't shaped quite properly. They'll be bent like this or this. And however they settle, they're always going to hit a tip. And so there is a non-coin toss element to selection for adhesion. Okay, here's another time lapse where we're watching the spores settle down towards us on an inverted microscope. And I'll let this play through again and hopefully pause it in just the right place. Well, so if I could get it paused in the right place, which I don't think I can. Oh, there we go. Okay. You can see the ConA reactive glucan strip. It's facing the same direction on those 2 spores, A and B. But as the movie plays, you can see the spores settle in opposite directions of each other. B is settling with ConA down and the spore attaches, and A is settling with the ConA reactive strip up and it does not attach. So it's the settling of the spore which determines whether it will attach or not. Okay, since this is a glucan, our presumption, because ConA binds to glucans, our presumption is that this is a glucan moiety. We decided to treat spores with an enzyme to determine whether that strip is the actual adhesive element. And so on the left, we see what's familiar to you now, this rotation of a spore. And it's our control where you can see the actin cytoskeleton as well as the ConA reactive glucan reactive strip. And to the right, we have used enzymes. We actually still have a stash of Novozyme in my lab. So that's our glucanase, is Novozyme. And you can see that ConA reactive strip has been removed from the spore with Novozyme treatment. And there's some debris in the field, and that's actually very lucky for this image because it shows us that the ConA is actually present in this preparation, because you can see the debris in the background reacting with the ConA, but the strip is entirely removed. So here is further confirmation that the strip is composed of, at least in part, of glucans. And now the image that you see to the right is of the glucanase treated spores. And I hope what you will see is that almost all of the spores in this image are unattached. So there are rare occasions in here where a spore looks to be immobile, like below this strip that's playing here is an immobile spore. But for the most part, these are all moving with the flow of liquid inside of the chamber that we're using here. So no ConA reactive strip composed of glucans, there's no attachment. And then that's quantified over here. Control is roughly 50% attachment. Enzyme treated, we get close to zero attachment. Okay, next we wanted to know, since the previous study I mentioned before from Bergstrom and Nicholson had showed that there is a secreted adhesive from Colletotrichum, we wanted to know the timing of the importance of this ConA reactive strip to attachment. And the way to get at that would be to fix the cells. And the data that I'm showing here are formaldehyde fixed cells. So the Conidia were treated with formaldehyde to kill them, and then attachment assays were performed. And it's indistinguishable of the percent attachment that's achieved when formaldehyde is used to kill cells versus living cells. So that to me indicates that the liberated spores that we harvested are already attachment competent. So this is not. Sorry, I got a battery low warning. I want to make sure I get this thing plugged in. I'm not sure why that would be. Yeah, my charger is not working for some reason. Well, hopefully this is gonna, hopefully I can finish up before my battery dies. Okay, next we wanted to see, can we flip spores and cause them to attach? And the answer is yes. So what we're using here is a micro controller that is controlling a motor that we got out of an old cell phone. So you're all familiar with turning your ringer off on your cell phone and somebody calls you and your cell phone vibrates. And there's a little motor in the phone that does that. And that we have attached to a slide here. And it's controlled by a microcontroller that controls the length of vibration, the time of vibration, as well as the intensity of the vibration. And in this image, what we're going to see is, you can see the vibration happen in the video as it occurs. And if we look across this field, you can see, for example, here is an unattached spore that flips over and becomes attached. Here is an unattached spore that doesn't flip over and it remains unattached. Here's an unattached spore that doesn't flip over, it remains unattached. Here's an attached spore that remains attached. But the punchline of this movie is seeing that spores can flip over and immediately attach upon that flipping. Sorry, I'm entirely distracted by my charger not working. Okay, we address this in another way by pipetting a solution of spores onto a substrate, allowing them to settle, quantifying, because we're working with an inverted microscope, quantifying attachment, and then after various time periods, using the pipetter to agitate the liquid solution. And so that's what we're seeing here. So no disturbance with a pipetter, we've got roughly 50% attachment as shown with this gray line here across the board. But if we allow them to settle and then disturb after 5 minutes, we achieve much higher percentage attachment, 80 to 90% attachment. If we allow them to settle for 10 minutes, it remains at roughly 50% attachment. And then 10 minutes in disturbance, you get nearly 100% attachment. And then here is allow them to settle and quantify for 15 minutes, and we remain at 50% attachment. And then it goes to a high level of attachment after the disturbance. So we can flip these spores, whether we use a cell phone motor or a pipetter, and they will attach. Okay, so what does all this mean? We think that the mechanism that we're seeing here is to enhance the dispersal of the fungus. So if all spores attached upon initial settling of the spores, then the size of the infection court would only be as big as the rain droplet that settled the initial spores. We think, again, this is about dispersal of the spores. So if spores were to settle and all attach in that rain droplet, then you're going to be dealing with only very small lesions at any one time. But the fact that only some spores will attach initially, then if the droplet of water containing spores runs down the leaf or falls off the leaf to another leaf or another plant, or maybe a subsequent raindrop splashes this, or maybe the rain droplet dries out and we have some unattached dried out spores that will then rehydrate and be dispersed again with a subsequent dew or rain event. All of these things can then result in further dispersal of the spores. This is all about then dispersal and selection for greater dispersal. We're still working on ways that we could quantify this. So one thing that we've tried is to look at cover slips. You know, I'm a benchtop scientist and a microscope scientist, so I think of ways of doing this artificially. So we looked at these long cover slips at various angles. And if we've got a flat cover slip, then we get about 50% attachment. If it's angled slightly, so the drop never rolls and all the spores are more or less distributed in the initial droplet. But if we've got a greater angle on the cover slip, allow the spores to adhere or to settle, then we get some spores in that initial droplet. And then of course, this is kind of expected, I guess, but as that droplet moves down the cover slip, you get deposition of spores down the cover slip as it runs. And so I'm still working on ways to model this. And we've tried to do this on corn leaves as well, but the microscopy gets exponentially more difficult to achieve if we try to do this on corn leaves. So then in conclusion, then we've got our Conidial Cointos model that we see here is now enhanced by also having the presence of that adhesive strip, presumably composed of glucans. And so we've got this polarized actin array that's pre-formed and pre-germinated Conidia, but we know that the production of that actin array occurs when the spores are liberated from the conidiogenous cells. The actin array tracks a discrete area of adhesive. We don't know yet if that actin array would be the distribution network that allows the secretion of that adhesive material, or is it actually more of a sensing mechanism? Since I also told you that these spores don't germinate unless they're attached. So the spores must sense in some way that they have become immobilized. And that actin array may also be participating in that sensing mechanism. So the initial attachment is passive. That adhesive is preformed and attachment occurs instantly upon contact with the surface. Attachment is sided. So this likely enhances spore dispersal. So outstanding questions. What are the relationship between the actin and the adhesive? I've just sort of mentioned this. Is the actin the distribution network? Probably yes, but also probably the actin is as part of the sensory network associated with the spore realizing that it's become immobile. What's the relationship between this and the previously characterized secreted adhesive? So I have a collaboration with some physicists that do Raman spectroscopy, and we're actually in the process of using Raman spectroscopy to try to determine what's in the adhesive. So how does that adhesive at the spore tips affect the coin toss? It isn't actually perfectly 50-50 attachment, and that's because of the adhesive that we see at the tips. And that's going to make modeling of this much more difficult. And I've got a epidemiologist who's helping me with the conceptualization of this. On the effect of wettability. So we've been using this mostly on cover slips. We don't know yet really the effect of this on a corn plant. And the corn plants are the leaves are going to be waxy because of the cuticular wax, and they're going to be more hydrophobic. And we don't know yet the effect of that on this dispersal mechanism. And I'm going to play this video. This is really a mystery to us. So we've got the actin array here. And this is using the dye FM4 that shows us a membrane within the cell. And I hope that what you see in this pre-germinated cell is the dynamic collapse of that actin cytoskeleton. And it's actually being achieved by this membrane-bound organelle that is trafficking on the actin cytoskeleton, almost like Pac-Man gobbling it up. And I've shown this to some of my colleagues who work on actin cytoskeleton. And we have yet to come up with a convincing hypothesis for what that structure might be that's actually causing the collapse of the actin cytoskeleton. But we do see this often enough in the cells to know that the dynamic collapse of the actin is actually happening amongst these cells in pre-germinated cells. And we don't know the mechanism for that at this time either. Okay, so finally then, I just want to mention again that my history has come full circle. So I was a grad student with Harvey Hoke, and I used to work on spore attachment. So here's a 1999 paper with me and Harvey, in which we were characterizing an adhesive sheath on the spores of the black rot of grape fungus. And I will also point out that this inquiry took us then to try to look at calcium signaling during that spore attachment. And we largely failed at that, but I'm pointing this out because I actually co-authored a paper with Gillian. Probably Gillian, you don't even remember this paper. All the way back in 2001, and my dear friend and departed colleague, Nick Reed, was also an author on this paper. Okay, so with that, then I will show you that these are the people that actually did the work. Joe Vaselli is a senior grad student in my lab who'll be graduating soon. He has fantastic microscopy skills and will be looking for a postdoc. If anyone up there needs a microscopy-oriented postdoc, he would be a good person to do it. Alan and Hope are two undergraduates who work on the project. And then Ali up here is brand new grad student who's going to be taking over from Joe on the project. Now, I think with that, then I can end and ask if there are any questions.
Outstanding job, Brian. Thank you so much. And questions?
Well,
I return to my comment, which you missed, Brian, due to your computer malfunction. Did you learn more from Chris at Michigan State or from Harvey Hoke while you were a graduate student here? Don't answer.
You know, I learned from both of them, so that's a loaded question. But, you know, I have focused on microscopy for most of my career, and
Yeah.
I certainly inherited that phenotype from Harvey.
Yeah.
I think
Stunning
my approach to
images.
this presentation was a very Harvey-like approach to a presentation. And
A serious question. Have you stained for DNA? in the matrix, perhaps, of those Conidia?
I have not. What would
Ah.
you expect to find?
Secreted DNA,
Yeah,
maybe.
okay. Well, we've tried to use DAPI because we were hoping to image nuclei as this happens to see when mitosis is triggered relative to germination. And DAPI doesn't work that well in live cells. And so really what we would need is a histone marker for that. That probably wouldn't help us with secreted DNA. I guess DAPI probably would work with that. I don't think I had that in mind when we looked at those DAPI stained spores. So I may have missed it if there was DNA in the secreted matrix.
Thanks.
Sure.
Gary?
Hey, Brian, I really enjoyed your seminar. Your microscopy just blows me away. A couple of things. For one thing, you solved the mystery that for many years working with that organism, when we're trying to optimize inoculum production, we looked for germinability, i.e. viability, using watch slides and looking at germination at 6 or 12 hours. And we're always frustrated, why couldn't we get that germination rate higher? So you've answered a great mystery there.
When we're trying to achieve nearly 100% germination, we will routinely deposit spores and then agitate it with a pipette.
Great.
And then you'll get nearly 100% germination.
Yeah, that's fundamental stuff. That's amazing. A couple of things I was thinking about as far as some of the implications, possibly, of some of your findings, and I agree with the dispersal thing. Two things occurred to me. To enhance the chances for stalk rot infection, enough of the spores need to get past the leaf blade and make it down behind the leaf sheath to be near the stalk rind. So, they don't all get bound up on the leaf blades. Maybe, I don't know, but maybe that would allow for more of the spores to get in behind the leaf sheath. That's just a thought. The other thing that occurs to me is that Ralph Nicholson did work with, found a germination self-inhibitor when the spores are all crammed together in that acervulus that probably are prevented from germinating. But the dilution factor, the idea that, a clump of spores landing in a droplet, some would adhere and some would be allowed to wash off, would be a dilution factor too, to kind of dilute out whatever germination self-inhibitors might be there. And that's a mechanism you might think about a little bit too, going along with dispersal. But anyhow, it was fascinating. I loved it, Brian. Thank you.
That's a really good point about the self-germination inhibitor being diluted as part of this coin toss mechanism as well. So I think I want to add that to the conceptualization of it, for sure.
Kathy?
Hey, Brian, nice to see you.
Hi, good to see you too.
I guess I always ask impossible questions. So I wanted to ask, like, how do you think this is like a kind of universal thing? Like all those fungi that have like curved spores, do you expect to find a similar mechanism?
I would not be surprised if we saw it with other fungi. How universal it is, I don't know. Okay, yeah, I think that it's not outside of, I don't think it would be wild thinking to think that fungi that cause foliar lesions that are water dispersed might have similar mechanisms. And for that matter, maybe even aquatic fungi. We think of fungi that float down a river and get snagged on leaf debris or whatever. I think anytime there's water dispersal, that a mechanism like this might be at play. Whether an aerial dispersed fungus, like the fungus I'm used to working on, Aspergillus nigilans, I'm not sure the mechanism would work similarly in a fungus like that.
Wow, it's so interesting and so beautiful. I love to see the sort of living fungus, because I usually just kill them when I look at them. So thanks for that.
Yeah, thank you.
Tristan?
Hi, Brian.
Hello.
I really enjoyed your talk, and your microscopy is absolutely stunning.
Thank you.
I don't know if I may have missed this during your talk, but I was really curious. So when Conidia are being liberated from their respective conidial spores, have you been able to observe any patterns in how the spores determine where their actin initially concentrates, whether there is a factor of light or initial orientation, or is that actin concentration more or less random?
Yeah, that's a great question. And we've actually tried to ask it. So back to my banana model, my camera's in a different place here. So really what the question is, I think, is the actin right or left-handed, right? So there is a sidedness to the points. There's the scar from the conidiogenous cell and there's the tip. So we can distinguish those. And we've tried to address, is the actin always made on one side or the other? And I'm not sure we've convincingly addressed it yet, but the preliminary data we have shows that it can be formed on either side. So there doesn't seem to be a preference of sidedness for which side it forms on.
All right. Well, I think many of us get to connect with Brian throughout the day, which we look forward to. And let's all thank Brian again for a really great seminar.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.